Hi everyone, I'm Tanya, and I just finished my second year here in the bioinformatics program here at UCLA. And today I'm super excited to share with you one of the projects I've been working with Kirk Miller on developing a method to detect whether recombination is mutagenic using genome-wide divergence dataset. So to me, one of the most important parameters in population genetics, especially in a lot of inferences projects, is mutation rate estimate. It is a key parameter to interpret genomic differences between different individuals and populations. It's also a key parameter to infer a lot of the evolutionary past events. So in the field, there's a lot of researchers who are interested in questions such as where in Africa humans come from, or where was the first domestication happens in dogs. And in order for any of these inferences projects to happen, you need a very accurate estimate of mutation rate. And so when talking about mutation rate, for example, when you ask what is a human mutation rate, so people often give you a number such as, I don't know, 2 times 10 to the minus 8. And that may give you the impression that mutation rate is constant across the genome. But that's not the case. In fact, mutation rate varies across the genome. So in population genetics, we use DNA differences between human and chimpanzee, for example, and that is called a divergence in order to use it as a proxy for mutation rate. So this figure is from a review paper in 2011, and what they did was they scanned along the genome looking at 1 megabase region at a time and compute the number of DNA differences between human and chimpanzee, and then plot the histogram of it. So what this histogram shows is that mutation rate is not constant and that it varies across the genome. But what the mechanisms that cause mutation rate to vary across the genome is not entirely known. There are several proposed mechanisms, such as replication time differs between different regions of the genome, or different regions of the genomes have different mutation rates or distance from telomere or GC content. But what I'm going to tell you about today is our project on understanding whether variation in recombination rate could explain away the variation in mutation rate. One of the hints that point us to looking at the variation in recombination rate is that we think that this could have some molecular biology basis. So recombination is initiated first by a double-stranded breaks. And when in the process of repairing these double-stranded breaks, these repairing mechanisms are actually quite error-prone. And so the idea is that at regions of the genome that have a lot of recombination, therefore a lot of double-stranded breaks happen, and then you accumulate more mutations. This was confirmed in experiments with yeast, and I think the next speaker will talk about more experiments in yeast to examine mutation variation, so that'd be interesting to know. But as population geneticists, we wanted to see whether there's evidence for recombination to be mutagenic in a natural population, such as in humans or in dogs, where we cannot do this type of experiments with. A second hint that points us to look at recombination variation is that we observe a positive correlation between DNA differences found between human and chimpanzee and recombination rate. So in population genetics, when we see this type of pattern, there's 2 hypotheses that can explain this pattern: natural selection and mutagenic recombination. So for the first part of my thesis, I examined whether natural selection can explain some of this correlation. And this one is currently in Project Plus Genetics, and I don't have time to go through this project today, but I'm happy to discuss with any of you about it afterward. But what we saw was that even though natural selection can explain some of these patterns, it can only explain the pattern at regions of low recombination. And so this led us to hypothesize that perhaps we need to invoke mutagenic recombination in order to explain the patterns at regions of high recombination. Well, the question of whether recombination is mutagenic is not new at all. There's other researchers in the field who have been working on this project for number of decades, but what they found is rather conflicting. There's a paper in 2014 that found that SNP density is elevated at regions of high recombination, and this is consistent with the hypothesis that recombination could be mutagenic. Then Franchioli et al. In 2015 did something really interesting. What they did was that they whole genome sequenced 250 Dutch trios that is comprised of a father, a mother, and a child, and that allowed them to calculate the de novo mutation rate. And then they found that the rate of de novo mutation is highly correlated with local recombination rate. But what's interesting is Pellamere et al. Used the same data set, and after they correct for the confounding effect of gene conversion, this effect went away. And so due to these conflicting results in the field, we wanted to see whether we could infer whether recombination is mutagenic. And we wanted to do this quite differently from what have been doing so far, which is that we want to use a genome-wide divergence data set. And we also want to correct for the confounding factor of natural selection. And so I wanted to talk a little bit more about why we use the genome-wide divergence data set. So current methods relies on trios data set that I talked about earlier that consists of a father, a mother, and a child. And for this type of data set, you need whole genome sequencing in order to call the de novo events because it's quite rare. And so you can imagine that this type of data set is not available, or not feasible for non-model species besides human. And so our hope is that if our inference method works, we could apply this to a variety of species that only require divergence data set, which is easier to obtain. So for the next few slides, I want to walk you through our model, how we do the inference. So in population genetics, we model the accumulations of mutations following a Poisson process written here, where K represents the number of mutations, and this is our data. Mu is the background mutation rate, and T is the genealogy. So what is this genealogy? Imagine that you have a human sequence and a chimpanzee sequence, and you want to know when in the past these two lineages coalesce. And so the paths that underlie these coalescent events are unknown, but we can get around these unknown genealogies by simulating a lot of these genealogies and integrate them out. So to simulate these genealogies, I apply the coalescent simulations that Sriram gave a really good tutorial on last week, and happy to discuss more about that too. So again, here's my Poisson process where I compute the probability of the data given the parameters. And I want to point out that in order to infer whether recombination is mutagenic, I've replaced mu, which is the background mutation rate, with mu plus phi r, where r is my recombination rate, and phi represents a coupling parameter between mutation rate and recombination rate. And here again, I mentioned that I want to simulate a lot of the genealogies and integrate them out, and then compute the probability of the data given the parameter following this Poisson process. So more specifically, I simulate 100,000 simulations to reduce the Monte Carlo variance. And I mentioned that I want to correct for the confounding effect. So this is taken into this step here when I simulate these genealogies. And then I compute the probability of the data given these parameters. And again, just to emphasize, the goal is to infer for a specific value of phi that can best recapitulate the data. So here's some of our early results. Here is, on the y-axis is the DNA differences found between human and chimp, plotting as a function of recombination. This is what I showed before, and this is our empirical data. And the first step that we did was to test whether a null model, a model that does not incorporate mutagenic recombination, to see whether this can recapitulate the data. And to do that, we forced phi to be equal to 0. And we saw that when phi is equal to 0, meaning there's no effect from mutagenic recombination, the simulated data cannot recapitulate what happened at regions of high recombination. But then when we allow phi to vary, when we infer for a specific value of phi, we saw that with a value of 0.014, now the simulated data can match pretty well with the empirical data. So that's pretty promising. And so what this means is that we saw that models that incorporate the effect from mutagenic recombination can better recapitulate the data at regions of high recombination. And what does the value of 0.014 mean? It means that a 1 unit increase in recombination rate as in the unit of centimorgan per megabase is associated with a 1.4 times 10 to the minus 10 increase in mutation rate in the unit of per base pair per generation. So all of this work that I've been trying to test with the human gene divergence data set is pretty new, and I wanted to test the method more rigorously using simulated data where we know what is the specific parameter is in order to compute statistics of interest such as confidence interval. And then as I alluded to earlier, I wanted to apply this method to a variety of non-model species in order to learn something interesting about the process of recombination other than human. So with that, I would like to thank my advisor, Kirk Lohmiller, a postdoc in the group, Christian Huber, who has helped me a lot with the inference project, and our funding agency. And thank you everyone for listening.